Hello everyone, welcome to Get My Class. So let's solve this question which says the introduction of dash was such that it generated both sense and antisense RNA in the host cells by using agrobacterium vectors. Option A RNA, option B protein, option C DNA, and option D lipid. Now let us understand the question. It says the introduction of dash was such that it generated both sense and antisense RNA in the host cells by using agrobacterium vectors. So first of all, we have to know what are these sense and antisense RNA. What do you mean by that? Sense and antisense RNA are nothing but the RNA molecules which are complementary to each other. They are complementary to each other. That's why they're called as sense and antisense. Now we know that RNA is formed from where? It is actually formed based on the information of the DNA. So we know the central dogma, the DNA will code for RNA, that is mRNA. Okay? And further, this mRNA will code for the protein. Suppose you have an organism which is causing the disease, and that disease is caused due to the production of a particular protein. Now, if we have the capacity to stop the production of protein, then we can stop the disease as well. Okay, so this will produce, actually this will cause, this is the reason for the disease. So if we stop the production of protein, we can stop the, you can say stop the disease as well, right? So what can we do? Because anyhow DNA to mRNA, there is no problem. But from RNA to protein, there is a problem. Because protein is the one which will cause the disease, right? So we can do one thing, we can block this mRNA in such a way that this does not code for protein. Okay. How can we block the mRNA? We know that double-stranded DNA, it will get separated as single-stranded, right? This is double-stranded DNA and it will get separated as single-stranded and each strand acts as a template, right? And for this, each strand will code for mRNA, right? And here, this is the DNA, you will get mRNA here, okay? Because it will be complementary. This is mRNA, okay? So again, here also it will be producing mRNA. Right? Now, what do we do here? So what I'm trying to say that the RNA molecules which are produced here, if they are blocked, okay, so RNA molecule can get translated only when it is in single-stranded, single-stranded form. But now what are we doing? We are taking the gene for this particular protein and inserting into the vector. Okay, it is the gene. So this is part of the DNA, right? Okay, and which is within the DNA vector. Now this will be like, this is nothing but the TI plasmid, that is tumor inducing plasmid, which has been taken from the Agrobacterium species. Okay, this will be introduced into the host where this DNA, which is having the gene for the protein will give rise to both the strands, that is sense strand and antisense strand. So what will happen? These strands will bind to the mRNAs which are produced here and they will form double-stranded RNA. Okay, double-stranded RNA. And this double-stranded RNA cannot form protein. It will not undergo translation. Okay? So that is why we can say that introduction of the gene, that is gene, when we are saying, we're talking with respect to the DNA, because genes, we consider genes which are part of the DNA itself, right? Okay, so let us see. The question says introduction of dash was such that, so introduction of DNA, okay, so DNA was such that it generates both sense and antisense RNA, because it is double-stranded DNA and it will definitely code for both sense and antisense RNA, which will go and whichever the complementary strands, they will go and combine with the RNA of the host. Okay, and further it will get blocked because the RNA molecules will get converted to double strand. Okay, in the host using Agrobacterium vectors. So A says RNA, no, we are not introducing RNA, we are directly introducing the DNA, because DNA will code for the RNA. Protein, no, we are not introducing. Yes, DNA. Lipid doesn't play a role at all. So let us conclude. The answer is option C. I hope you understood the explanation. Thank you, everyone.